everyone. I'm Apoorva Shankar, Assistant Professor from Department of Life Sciences, Krishna Jayanti College. Today, let us discuss the basics about microscopy, the Brightfield microscopy, and the principle of Darkfield microscopy. So these is the basic parts of a light microscope. So here you can see from the top, we have an eyepiece, which has an ocular lens. And then you have the rough adjustments or the coarse adjustments which can be done for the body tube. And then we have the fine adjustments. And we have the nose piece, which has the different types of objective lenses. We have usually three different types of objective lenses. One is for the oil immersion lens, and then we have the lower objective lens, and then the medium power objective lens. Then you have a place for the stage where you can keep your slides, followed by the condenser, the iris diaphragm, and the light source. Here we have the mirror for directing the light source, and then we have the base of the microscope. So these are the basic parts of a normal light microscope. So we have the different mechanical parts here. We have this metal stand, which is a supporting part of the microscope, which gives the stability to the instruments. It consists of the base, the pillar, the curved arm, which is attached to the pillar. The light source is usually at the base, and it can be an inbuilt electric lamp or a mirror. The mirror can be adjusted so as to get the maximum light to fall on the object to be viewed. Usually the mirror that is used for the microscope has two types of surfaces. The plane side, which is used for the artificial light, and you have a concave side, which is used for the natural source, that is the sunlight. Then we have the iris diaphragm. The iris diaphragm, which is present below the stage, it helps in adjusting and regulating the amount of the light that enters into the condenser. So for oil immersion microscope, the objective lens or this diaphragm is fully opened. For high power objective, the iris diaphragm is partially closed, and low power objective, it is almost closed. So this closing of the iris diaphragm is very important for the different objective lens because it controls the amount of the light that is entering into our ocular lens. So for oil immersion lens, it is fully open because the space between the oil immersion lens and the specimen under observation is very less. So it is important that the maximum light enters into the objective lens and into our ocular lens. And for higher objective, it is partially closed because the distance between the specimen and the higher objective lens is minimal. And for lower power objective lens, the distance between the specimen and the objective lens is higher. So it has to be closed to regulate the amount of the light that is entering into the ocular lens. So the next one is the stage, is the platform where we use our sample for observation, where we keep a specimen which is in the slide for observation. So this is allowed in such that it has an opening in the center that allows the light from the condenser to fall onto the objective lens. The stage is attached with a clip which helps in moving the slide in the horizontal and vertical directions in order to scan the specimen throughout. So next we have the body tube or the barrel. This is attached to the curved arm of the microscope. The body tube can be moved vertically with the help of the coarse and the fine adjustment knobs. These knobs are used to bring the object into the focus. At the lower end of the body is the objective lens, and at the upper end is our ocular lens. So the objective lens system, as I told you earlier, there are three different types of objective lens systems which we have to view the specimen. One is a lower power objective lens, which has a magnification up to 10x. And the focal length, that is the focal length is the distance between the objective lens and the slide or the specimen. So this is called the focal length. The focal length is usually 16 millimeter for the lower objective lens. And then we have the higher objective lens, power objective, which is up to 45x. Here the focal length is 4 millimeter, which means the distance between the objective lens and the specimen is about 4 millimeter. Then we have the oil immersion objective, which has the magnification power up to 100x, and the focal length here is very less. That is, the distance between the specimen and the objective lens is less than 2 millimeter. So based on this, that is the distance, that is the focal length, we have learned by now that the iris diaphragm should also be adjusted accordingly. So for lower power objective lens, the iris diaphragm is almost closed. For higher power objective, it is partially closed. And for oil immersion objective, it is completely open in order to allow the maximum amount of the light to enter into the ocular lens. So these three lenses are mounted on the revolving nose piece. Then we have the oil immersion objective. So in the oil immersion objectives, the other two lenses are actually the dry lenses, that is the low and the higher power objective lens. Whereas the oil immersion objective requires a special kind of oil to be between the specimen and the objective lens. And this helps in achieving higher magnification. So what is the kind of oil? We cannot use any oil for an oil immersion objective. It is very important that the oil that is used for the microscopy should have the same refractive index of that of the glass slide that is used. That is 1.52 is the refractive index that has to be used, so that it provides a homogeneous path for the light to travel. So this is very important. The refractive index of that of the oil used and that of the glass side should be same. So we all know that the behavior of the light. So when the light passes from a rarer medium to the denser medium, what happens? So when one medium, we all know that there is the behavior changes of the light when it passes from one medium to another. So when the light travels from a rare medium to a denser medium, it bends towards the normal, which you can see in the image here. Whereas when the light travels from a denser medium to a rarer medium, it bends away from the normal. So this is the principle that is used in the oil immersion objective lens when we are using the oil. So when we have an air in between the objective lens and the specimen, the light rays that is passing out from the glass slide, which is the denser medium, to the air, which is the rarer medium, it bends away from the normal. Hence, many of the light rays do not enter into the objective lens. So when we use oil in the medium, which has the same refractive index as that of the glass slide, so it does not bend away from the normal. As a result, leaving the objective, it will travel straight into the objective lens. So this is the principle of using the oil. The oils that are generally used are cedarwood oil, Canada balsam oil, xylol balsam oil, and glycerin can be used. We can see that all the refractive index of these different types of oils lies between 1.4 to 1.5, which is exactly the refractive index of the glass slides which are used for the study. The next we have the ocular piece or the eye lens. This is present in the upper end of the body, consisting of lenses which will further magnify the real image that is produced by the objective lens. And usually the magnification of the eyepiece is around 10x to 15x. The next part of the microscope is the condenser, which is present above the iris diaphragm. The condenser consists of two sets of convex lenses that concentrate and intensify the light that is reflected by the mirror. So in the types of microscope, so we discussed about the light microscope and its parts. So we all know that in the light microscope, when we observe the specimen, we see the specimen as darkly stained specimen against the bright background. So that is our Brightfield microscopy. Now let us look into the Darkfield microscopy. So in Darkfield microscopy, we see a bright image of the object against a dark background. So this Darkfield microscopy can be used to study the living, and the unstained preparations can be studied using this type of microscopy. So as we can see in the image here, you can see a dark background and an illuminating cells which are unstained, and light and illuminating cells can be seen here. So in the dark field microscope, the light source can be a bulb. The condenser is what is different in a dark field microscopy. This condenser is used to block most of the light from the source, and it contains, how does it do this? It does that with the help of the annular patch or the annular stop, which is present at the center of the condenser. So this results only in the cone of light which hits the specimen, keeping the background dark. So this is the working ray diagram of a dark field microscopy. Here, as you can see, there is a dark field stop or an annular stop which is present in the condenser. This annular stop does not allow the light to pass through the center of the microscopy and allows only the light rays to pass through the corner of the lenses. And this type of condenser is called an Abbey condenser, which produces a dark background. So this light passes through the corners of the convex lenses and the condenser and hits the specimen. And the light that is given out by the specimen passes through the objective lens to form an image. And the light of the background does not enter into the objective lens. So that is why the background light is omitted, and the background appears dark in color, while the specimen, the light which is reflected by the specimen, only enters through the objective lens, and that is why you see a bright specimen against a dark background. And the condenser is the one which does the job here, and it is called an Abbey condenser. It has a dark field stop or an annular stop at the center, which prevents the light from entering through the center and hitting the specimen. So this is a dark field microscopy. The living and unseen microorganisms can be observed by simply changing the way in which they are illuminated. So here the cone of light is focused on the specimen in such a way that the unreflected and the unrefracted rays do not enter into the objective lens. Only the light that has been reflected or refracted by the specimen forms an image. The field surrounding the specimen appears black, while the object itself is brightly illuminated. So because the background is dark, this type of microscopy is called a dark field microscopy, and it is considerably internal structures are often visible, and larger eukaryotic microorganisms can be studied by this type of microscopy. The dark field microscopy can be used to study the causative agent of syphilis, that is Treponema pallidum. So the applications, the dark field microscopy, in contrast, is created by the bright specimen on the dark background. It is useful for studying the outer outlines, the edges, the shapes of the various cells, and it does not provide greater information about the internal structures. The dark field can be used for studying the marine organisms such as algae, planktons, diatoms, insect fibers, hair, yeast, protozoans, and also the structure of certain minerals, crystals, polymers, and ceramics. It is also used in the research of the live bacterium can be studied. It is useful for examining the outline details, and not much information is provided about the internal structure. So this is about the dark field microscopy. So in comparison with the light field microscopy and dark field microscopy, the condenser is the one which is differing. Here in bright field microscopy, the condenser allows the light to pass through the center of the specimen, and also the light which is reflected and unreflected also enters into the objective lens. Whereas in the dark field microscopy, the light that is only reflected and refracted by the specimen enters into the objective lens and goes out to form the image. And that is why we see a bright image and a darkly stained specimen in the case of bright field microscopy, whereas in case of dark field microscopy, we see a dark image, dark background with the illuminating cells. Thank you. I hope the concepts are clear now. Thank you.